Muscle biopsy studies are the gold standard of creatine research, and understanding how they work helps you interpret the evidence behind everything else claimed about creatine supplementation. Most sports nutrition claims rest on blood tests, performance measures, or questionnaire data. Creatine's claims at their foundation rest on actual muscle tissue samples, showing what's happening inside the muscle cells. A muscle biopsy involves a small needle or incision into the muscle belly, typically the vastus lateralis, the large outer quadriceps muscle ear, and extraction of a tissue sample roughly the size of a grain of rice. The sample is frozen immediately to preserve its biochemical state. It's then analyzed in the laboratory for creatine content, fiber type composition, satellite cell numbers, myosin heavy chain isoforms, enzyme activity levels, and numerous other parameters depending on the research question. The direct measurement of muscle creatine content through biopsy is what established the basic facts of creatine pharmacology. Before biopsy studies were done, researchers could only infer what was happening in muscles from blood tests and performance outcomes. Biopsy studies in the 1990s directly demonstrated that oral creatine supplementation raises muscle creatine content by exactly the amount needed to explain the measured performance improvements. This direct chemical verification elevated creatine from an interesting hypothesis to an evidence-based intervention. Subsequent biopsy studies have established the saturation ceiling, the rate of loading with different doses, the differential uptake between fiber types, the inter-individual variation in response, and the washout rate after stopping supplementation. Every major claim about creatine's mechanism in muscle is backed by biopsy data, not just functional outcomes. This is what makes the creatine evidence base uniquely robust. You can't say the performance improvements might be due to placebo when you have direct chemical measurement of increased muscle creatine content corresponding precisely to the performance gains. Biopsy studies have also been central to the satellite cell research on creatine. Immunohistochemical staining of biopsy samples using antibodies that mark specific proteins allows researchers to count satellite cells, measure their activation state, and track changes over time. The biopsy evidence for creatine-enhanced satellite cell proliferation is direct and histological, not inferred from downstream outcomes. For athletes and researchers evaluating any supplement claim, the question of whether biopsy data exists, whether we're looking at actual muscle tissue changes or just downstream performance correlations, is one of the most important quality filters available. For creatine, the biopsy data is extensive, replicated across research groups, and internally consistent. The mechanism is not hypothetical. It's been seen, measured, and photographed in human muscle tissue samples across hundreds of studies over 30 years. The biopsy research on creatine has also produced findings that extend beyond pure muscle physiology, specifically in the characterization of connective tissue changes associated with creatine supplementation and training. Some biopsy studies have included sampling of the tendon-muscle interface and paramecium, the connective tissue that surrounds and organizes muscle fibers. These analyses suggest that creatine supplementation combined with training may support a more robust connective tissue response with greater collagen synthesis markers compared to training without supplementation. The mechanism is not well characterized, but cell volumization's activation of mTOR may have downstream effects on fibroblast activity and collagen turnover within the muscle's connective tissue framework. Tendons and muscle connective tissue are rate-limiting factors in strength development. The contractile ability of muscle often advances faster than the connective tissue's capacity to transmit that force to bone. If creatine supports connective tissue adaptation alongside muscle fiber growth, the practical benefit is a more balanced structural development that reduces injury risk as training loads increase. This is preliminary data, and the effect size is modest compared to the muscle fiber changes that are creatine's primary documented mechanism. But it's a reminder that biopsy studies continue to reveal dimensions of creatine's biology that performance measures alone can't capture, and that the full picture of what this molecule does in trained muscle tissue is still being written. Biopsy methodology continues to improve, with advances in mass spectrometry and single-cell RNA sequencing beginning to allow more detailed molecular profiling from smaller tissue samples.